Hi everyone! So this video answers the question, what are the ways the brain protects itself from injury and or stress? By the end of this lesson, you should be able to answer that question using these vocabulary. So the content here is we're going to talk about how the brain protects itself from physical injury using the skull, the meninges, and the cerebrospinal fluid, how it protects itself from chemical injury using the blood-brain barrier, and how despite those things, there are still dangers to the brain from things like alcohol. Let's start with physical protection of the central nervous system. So, as you'll find out when you do your brain dissection, nervous tissue is very soft and delicate, and injury to neurons can be catastrophic because those neurons are irreplaceable. So if you damage your skin, you're just going to grow new skin cells because your skin cells will go through mitosis and replace the ones you lost. If you damage your muscles from working out too hard, well, those tears in the muscles would trigger muscle cells to go through mitosis to then grow new muscle. If you break a bone, even your bone cells, which are not as good at healing themselves, but even your bone cells can heal as long as you set the bone the right way. But there's a point in your childhood where your neurons can't really be replaced. They're not undergoing mitosis anymore, so they're amitotic. And that means that if you have damage to your brain or your spinal cord from something like a car accident, that damage is going to be permanent to a degree. Now, other neurons near the place where your brain cells have died, other neurons can reach out and try to form new synapses, new connections with other neurons around the area to try to recover some of your lost function, but it's never going to be a perfect replacement for the neurons that you lost. So there are three ways that your brain is physically protected. The first is you have your bone in your skull and in your vertebral column, right? So all the bony vertebrae that surround your spinal cord. And here's your spinal cord depicted right here. You have your meningeal membranes, which are the layers underneath the skull that basically are sandwiched between the skull and the brain itself. And then you have the cerebrospinal fluid, which is shown here in blue, just kind of circulating around the entire brain and the spinal cord. Let's start with the meningeal membranes, because you already know what the skull is. So let's move to the membranes. There are three connective tissue membranes, which we call meninges or meningeal membranes, that cover and protect the central nervous system. The topmost layer under the skull, here's your skull, right? So here's your scalp, here's your skull. And then underneath that, the topmost layer is composed of two sublayers, which we're not going to get into for my class at Pacifica. I will never ask you for the names periosteal and meningeal dura mater. But you do need to know that together they form a very tough fibrous layer under the skull called the dura mater. Dura is Latin for tough, and mater is Latin for mother. Next, the middle layer is this purple one here. It's called the arachnoid mater, and arachnoid means spider. The reason is you can see that it has these projections that go down and meet that layer below it in blue, and these projections create the space between the arachnoid mater and the next layer. And then the projections that kind of like create that space make it look like a spider. On top of that, you'll also notice that there are these arachnoid villi right here, these little like puffy thingies that are coming out from where the arachnoid space is, and I'll get into that later. Okay, and then the last layer is the pia mater, which you guys can see is this blue layer, and it clings to all of the sulci and gyri of the gray matter of your brain and your spinal cord, and it just is this gentle but clingy layer that adds yet another layer of protection for the brain. When you do your brain dissection, you shouldn't bother to try to peel off the pia mater because it's going to be tightly clinging to the surface. You'll notice, by the way, that the space between the arachnoid mater and the pia mater, this is called the subarachnoid space, sub meaning under. So the subarachnoid space right here is going to house the blood vessels, which you can see here, And then in addition to that, it's also going to be where the cerebrospinal fluid can circulate around the brain. So what is cerebrospinal fluid? It's a watery broth that's similar to blood plasma, and it's constantly being formed by your choroid plexuses. So you have one choroid plexus in your epithalamus, and then you have some other ones in other places. And the cerebrospinal fluid is made of a little protein, a lot of vitamin C, a lot of ions, a lot of water. It's always circulating among the ventricles, canals, and aqueducts in the brain. And if you ever have to get a spinal tap done, what they're essentially doing is they're putting a long needle, hollow needle, into the space around your spinal cord where the cerebrospinal fluid is circulating so that they can remove some of that cerebrospinal fluid from your lower back, from the region of the spine in your lower back, and they can look for things like toxins or other chemicals that might indicate that you maybe have an infection that could compromise your brain or your spinal cord. So here, this space right here where that blue fluid is flowing through, that's the subarachnoid space. It's sandwiched between the arachnoid mater and the pia mater that clings to the surface of the brain. And it is created by these red regions, the choroid plexus here and the epithalamus, plus the one in the ventricles over here and over here. So they're constantly producing the cerebrospinal fluid by basically taking water nutrients from your blood and cleaning it up and then sending it out to circulate around the brain, through the ventricles, around the cerebellum, down past the spinal cord and up again, around the brainstem, around the pituitary, and so on and so on. Now you'll notice that there are places where the cerebrospinal fluid does escape, and that would be right here and right there. These places are called arachnoid villi. And so essentially, you don't want the cerebrospinal fluid to just like stay there forever because that can encourage bacterial growth. And so as yet another way to protect itself, the brain is constantly creating new cerebrospinal fluid and draining the old cerebrospinal fluid. Speaking of arachnoid villi, remember this. These are the arachnoid villi. So these are one-way valves that let old cerebrospinal fluid out to go, back into the veins to then return to the heart, so that you're constantly just, circulating the fluid around your entire central nervous system. Here you can see the view of the ventricles. So these are also filled with cerebrospinal fluid that are circulating in and around. Here is the anterior view, so you can see here are the lateral ventricles here and here. Here's the third ventricle, so this is, 1 and 2. Here's the third ventricle, here's the fourth ventricle. Here's the side view of that. So you can see you have the lateral ventricle here, and another one on the other side. Here's the third ventricle, and here is the fourth ventricle. They are all filled with cerebrospinal fluid. When you do your brain dissection, you should be able to clearly see most of those ventricles. Okay, so we talked about how the skull, the meningeal membranes, and the cerebrospinal fluid give physical protection. The blood-brain barrier is also there to provide protection from chemical threats to the brain. So the brain cannot handle even the tiniest fluctuations of chemicals the way other organs can. And for that reason, your neurons are kept separate from the blood-borne substances in your blood that could compromise the brain by what we call the blood-brain barrier. So let me just show you what that looks like, and then we'll go back into what kind of substances are prevented from reaching your neurons. So in a normal blood vessel somewhere else in your body, this is what that might look like. Here would be one blood cell that makes up the wall of the blood vessel. Here would be another one, and then there would be these pores in between the cells that allow for stuff to go in and out, if it's a capillary, which are the smallest types of blood vessels. In the brain, however, the blood vessels, first of all, are tightly joined together so that things cannot easily go in or out. And then second of all, they're surrounded by all of these glial cells, these neuroglia, that basically just filter out anything that does come out from the blood, and kill off or destroy anything escaping from the blood that could be dangerous to the neurons. And so you can see here's the blood vessel again. Here's a capillary. Blood is flowing through this lumen here. And then you have this glial cell here called an astrocyte that basically prevents toxins or bacteria or other things from escaping the blood and reaching the brain. Here's another view of that. So this would be the blood up here in the tan region. And then here would be the cells that make up the blood vessels right here. These things here are the white blood cells that are there to also, within the blood, destroy things that could threaten your brain. But then on the other side, outside the blood vessel, you have these neuroglia. So you have the astrocytes, which, basically just filter out whatever might come out of the blood that shouldn't be there. And then you have these microglia as well that patrol the region and that eat and destroy any things like bacteria or whatever that might be escaping from your blood. So this is called the blood-brain barrier. What can get through the blood-brain barrier? The stuff that the brain actually needs. So stuff like water, glucose, essential amino acids, fats, respiratory gases like oxygen, and then some things that the brain doesn't need, but because of their chemical structure, they're also able to pass through. So fat-soluble alcohols, nicotine, caffeine, and anesthetics can also pass. Essentially, if it's a drug that will affect the way that you think or affect, like how your brain is working, then obviously it's something that can pass the blood-brain barrier. What doesn't pass? Metabolic wastes, the things that basically make up your urine. Toxins, proteins, and most drugs are prevented. So if you take, drugs for treating, like other conditions, most of those will not affect the brain. And then you also have non-essential amino acids and potassium, which are just always pumped from the brain in general. Last of all, we need to talk about how, despite all of these layers of protection, because of their chemical structure, there are some things that can cause damage to the brain, and it can be irreversible. So obviously, a concussion from playing football can cause brain damage, or a car accident can cause brain damage. But alcohol and other drugs or substances can cause brain damage, and it can be irreversible. So here you can see two images from a study that was done looking at teens who were admitted to binge drinking regularly compared to teens who admitted to drinking occasionally and in moderate amounts, and then also compared to teens who said that they did not drink. And what the researchers found was that there were significant differences in the brains of teens who regularly would binge drink compared to those other groups. And some of those differences were in the white matter. So you can see these red specks showing damage to the white matter. If we have time in class, I'm going to ask you guys to think about that and look through some evidence and have a group discussion. But for the remainder of this lesson, here are your key takeaways. When we talk about protection of the brain, you need to know that the brain must be protected because damage to the brain is usually irreversible. The skull, meningeal layers, and cerebrospinal fluid protect the brain from physical injury. The blood-brain barrier protects the brain from chemical injury. But despite these protections, the brain is still vulnerable to damage from trauma, like concussions, from toxins, like alcohol, and then from general degradation from things like old age and Alzheimer's disease. And do keep in mind that exposure to toxins like alcohol during your teen years can cause long-lasting changes to brain structure and function. I'd like you to go back to the first slide and just see if you can pause the video and answer this essential question using this vocabulary. And if you can do that comfortably, that means that you have mastered this topic.